Malaria is a disease that has shaped human history for millennia. Responsible for millions of deaths and countless suffering, malaria is one of the most significant infectious diseases, affecting millions of people across the planet each year. Today, we explore the science behind this deadly parasite, its impact, and the ongoing fight for a vaccine. Welcome to Malaria, fighting the world's deadliest parasite. Malaria is a life-threatening disease caused by parasites of the genus Plasmodium. It is transmitted through the bite of infected female Anopheles mosquitoes. Among the 5 species known to infect humans, Plasmodium falciparum is the deadliest, responsible for the majority of severe cases and fatalities. Malaria is preventable and treatable, yet it remains a significant public health burden, especially in sub-Saharan Africa, where 94% of malaria deaths occur. In 2022, an estimated 247 million malaria cases occurred worldwide, leading to over 619,000 deaths. While the global burden of malaria has decreased significantly over the last 2 decades, due to control measures like insecticide-treated nets and indoor residual spraying, resistance to anti-malarial drugs and insecticides poses growing challenges. Children under 5 and pregnant women are the most vulnerable, with severe malaria causing complications such as cerebral malaria, severe anemia, and organ failure. Understanding malaria requires a look at the complex life cycle of Plasmodium. When an infected mosquito bites a human, sporozoites are injected into the bloodstream and rapidly travel to the liver within minutes. There, they mature and multiply into thousands of merozoites through a process of asexual reproduction. These merozoites burst from liver cells and enter the bloodstream to invade red blood cells, where they continue to replicate. Inside red blood cells, the merozoites develop into trophozoites, which consume hemoglobin and grow into mature trophozoites, often forming a characteristic red blood cell ring stage. The mature trophozoites divide to form schizonts, which rupture the red blood cells, releasing more merozoites into the bloodstream to continue the cycle of infection. This continued and coordinated replication causes red blood cells to rupture in waves. Each cycle of red blood cell destruction releases toxins and leads to the hallmark symptoms of malaria, fever, chills, and anemia. In severe cases, infected red blood cells become sticky, leading to blockages in blood vessels, particularly in the brain, causing cerebral malaria. Some trophozoites differentiate into sexual forms called gametocytes, both male and female, instead of continuing asexual reproduction. These gametocytes circulate in the bloodstream and are taken up by a mosquito during a blood meal, where they undergo further sexual development. The sexual stages of Plasmodium are essential for the parasite's transmission between humans and mosquitoes. Malaria presents with a range of symptoms depending on the severity of the infection. Early signs often mimic flu-like symptoms: fever, headache, muscle aches, and nausea. As the disease progresses, periodic fever spikes, chills, and profuse sweating become more characteristic. Severe malaria can manifest as organ failure, cerebral involvement, respiratory distress, or severe anemia. Immediate diagnosis is therefore critical. The gold standard for diagnosing malaria remains the microscopic examination of stained blood smears. However, rapid diagnostic tests, or RDTs, are increasingly used, especially in resource-limited settings, as they detect specific antigens of Plasmodium within 15 to 30 minutes. Treatment for malaria varies by species and severity. For uncomplicated malaria, artemisinin-based combination therapies, or ACTs, are the first line of treatment. Artemisinin is derived from the plant Artemisia annua and rapidly reduces parasite biomass. However, resistance to artemisinin and partner drugs has emerged, particularly in Southeast Asia, complicating treatment strategies. In cases of severe malaria, intravenous or intramuscular artesunate is preferred due to its rapid action in reducing parasite burden. For non-falciparum species like Plasmodium vivax, treatment includes chloroquine or ACTs, often combined with primaquine to eliminate dormant liver stages, known as hypnozoites. Malaria control faces significant hurdles due to rising resistance. Resistance to insecticides used in mosquito nets and indoor residual spraying, particularly pyrethroids, threatens the efficacy of these interventions. Meanwhile, resistance to antimalarial drugs, most notably artemisinin and its derivatives, poses a major risk to treatment efficacy. Novel vector control tools are in development, including next-generation insecticide-treated nets and the use of genetically modified mosquitoes to reduce mosquito populations. However, sustained investment in research and innovative strategies will be key to future success. For decades, scientists have sought a vaccine to prevent malaria, but the parasite's complex life cycle and ability to evade the immune system have made it an elusive goal. The breakthrough came in 2021, when the World Health Organization recommended widespread use of the RTS S-01 vaccine, the first vaccine proven to provide partial protection against malaria in children. The RTS vaccine targets the circumsporozoite protein on the surface of sporozoites, preventing them from entering liver cells. In large-scale trials across sub-Saharan Africa, the vaccine reduced severe malaria cases by about 30% in children. Another promising vaccine, R21 Matrix M, is currently in Phase 3 clinical trials. This second-generation vaccine, also targeting the circumsporozoite protein, has shown up to 77% efficacy in early trials. It could represent a significant improvement over the RTS vaccine, providing higher and longer-lasting immunity. In addition, transmission-blocking vaccines are in development, which would aim to stop the parasite's spread by inducing immune responses in humans that prevent mosquitoes from becoming infected. Prevention remains the cornerstone of malaria control. Insecticide-treated bed nets and indoor residual spraying are critical in reducing mosquito bites. In addition, chemoprevention, such as intermittent preventive treatment in pregnant women and seasonal malaria chemoprevention for children, offers a proactive approach to reducing malaria cases in high-risk groups. Emerging strategies include gene-drive technologies that aim to suppress mosquito populations or alter their ability to transmit Plasmodium. However, these strategies are still in development and require extensive testing to ensure safety and efficacy. Despite the progress made, malaria continues to demand novel solutions. Advanced genomic techniques are being applied to study mosquito populations, identify new drug targets, and better understand the parasite's biology. Research is focusing on next-generation anti-malarials. These are new classes of drugs, including spiroindolones and imidazolopyperazines, which are being developed to combat drug-resistant strains. Host-directed therapies aim to enhance the host immune response to malaria, preventing severe disease by reducing inflammation and endothelial dysfunction. Artificial intelligence is being used in epidemiology research. AI models are being used to predict outbreaks, optimize resource allocation, and improve intervention strategies, offering more precise and cost-effective methods of malaria control. Vaccines have long been considered one of the most effective public health tools in the fight against infectious diseases. In the context of malaria, developing a successful vaccine has been a complex challenge due to the parasite's intricate life cycle, immune evasion tactics, and its ability to rapidly change forms within the human body. Malaria vaccines are designed to target different stages of the life cycle, from the sporozoites injected by mosquitoes to liver-stage parasites, blood-stage merozoites, and even the gametocytes responsible for transmission to mosquitoes. Let's take a closer look at the latest vaccine breakthroughs and trials. The RTS S&R21 vaccine, developed by GlaxoSmithKline and supported by global partners like the Malaria Vaccine Initiative and the Bill and Melinda Gates Foundation, represents the first ever malaria vaccine to reach large-scale implementation. It targets the sporozoite stage by stimulating the production of antibodies against the circumsporozoite protein, a protein essential for sporozoite invasion of liver cells. In Phase 3 clinical trials involving over 15,000 children across 7 African countries, the vaccine showed a 30 to 40% reduction in clinical malaria cases and a 29% reduction in severe malaria cases in children over a period of 4 years. These results may seem modest compared to vaccines for other diseases, but given malaria's immense global burden, even partial protection can have a significant public health impact. Importantly, in 2021, the World Health Organization recommended the widespread use of the vaccine in regions with moderate to high malaria transmission, particularly in sub-Saharan Africa. Pilot implementations in countries like Kenya, Malawi, and Ghana have already vaccinated hundreds of thousands of children, offering a glimpse of the vaccine's potential on a larger scale. While marked a major milestone, the search for higher efficacy vaccines continues. The R21 Matrix M vaccine, developed by the Jenner Institute at the University of Oxford, is showing tremendous promise as a next-generation malaria vaccine. Similar to the RTS vaccine, the R21 vaccine targets the circumsporozoite protein, but its design includes a larger antigen payload and a different adjuvant, the Matrix M, which enhances the immune response. In a Phase 2 trial conducted in Burkina Faso, the vaccine demonstrated up to 77% efficacy against clinical malaria, a significant improvement over the first-generation vaccine. Phase 3 trials involving 4,800 children in 4 African countries are currently underway, and preliminary results are highly anticipated. If successful, it could become a game-changer, providing higher and longer-lasting protection against malaria in endemic regions. With its simpler manufacturing process and potentially lower cost, it is also more scalable for widespread use in low-resource settings. Another exciting frontier in malaria vaccine development is the exploration of transmission-blocking vaccines, or TBVs. Unlike traditional vaccines that protect individuals from getting infected, TBVs aim to interrupt the malaria transmission cycle by targeting the parasite in its gametocyte stage, the form that infects mosquitoes when they bite a human host. One candidate, known as PFS25, targets a protein expressed on the surface of malaria gametocytes. preventing them from maturing inside the mosquito midgut. By blocking the development of infectious parasites within mosquitoes, TBVs could reduce the overall spread of malaria in endemic communities. These vaccines are not necessarily designed to protect the vaccinated individual from malaria, but rather to reduce community-wide transmission. When used in combination with other malaria control strategies, including RTS S or R21, transmission-blocking vaccines could play a critical role in malaria elimination efforts. Despite the progress in vaccine development, significant challenges remain. The complexity of the malaria parasite, with its ability to rapidly evolve and evade immune detection, continues to make vaccine design difficult. While RTS and R21 show great promise, their efficacy is still far from the levels achieved by vaccines for other infectious diseases like measles or polio. Researchers are now exploring multi-stage and multi-antigen vaccines that target different points in the parasite's life cycle. This strategy aims to induce broader immune responses capable of blocking infection at several stages, from sporozoite entry into the liver to blood-stage parasites causing disease. Other promising approaches involve leveraging cutting-edge technologies like messenger RNA or mRNA platforms, which gained attention during the COVID-19 pandemic. In addition to efficacy, there is the challenge of affordability and distribution. Malaria endemic regions often face significant resource constraints, making the widespread delivery of vaccines logistically challenging. Nonetheless, the global push for equitable vaccine distribution is critical to ensuring that the benefits of these advancements reach those most in need. The development of effective malaria vaccines represents one of the most important breakthroughs in the ongoing battle against this ancient disease. With RTS already saving lives and the promise of R21 and transmission-blocking vaccines on the horizon, we stand at a pivotal moment in malaria prevention. However, achieving the goal of eradication will require sustained global efforts in research, funding, and vaccine distribution. As science advances, so too does our hope for a future where malaria is no longer a threat to millions. The road ahead is challenging, but with each new vaccine trial, we move one step closer to a world without malaria. Malaria is a formidable adversary, but with continued innovation in vaccines, treatments, and vector control, we are edging closer to a world without this ancient scourge. Thank you for joining us in this exploration into malaria and the potential of vaccines. If you like this video, please share and remember to subscribe to Science Curious.